Excellent, here we go. So, yep, I'm gonna present the profiling results for the second round. So just a quick refresher about what profiling is. So basically we're taking the raw metagenomic reads over here and then using some sort of reference database, some sort of information or taxonomy information. And then from that, you want to get a taxonomic profile. So this over here is a CAMI formatted taxonomic profile, but basically what it's doing is it's telling you who is there and how abundant those different organisms are. So basically what it'll allow you to produce is one of these much hated little pie charts right here. But the takeaway is it's telling you who is there and how abundant they are. Quick recap on the data. I know this has been mentioned before, but there's marine data with 10 samples simulating a seafloor environment, and then 50 gigabytes in total for both short and long reads. The strain madness, we had 100 samples with a whole lot of strain level variation, and 200 gigabytes total for both the short and the long read samples. In this talk, I'm mainly going to be emphasizing the marine data here just because it's only 10 minutes, so you can only say so much. But what's important to say is what metrics we're using. So first off, since profiling tells you who is there, we are concerned about binary metrics, so in particular presence-absence metrics. So for each taxonomic rank, we have our gold standard because we already know what's in the sample because it's from simulated data, and each tool makes a prediction. So you have false negatives, true positives, and false positives. We consider a lot of binary metrics, but for this talk, for sake of time, I'm only going to be considering purity and completeness, aka precision and recall. So that was who was there. Next is and how much, and this is the relative abundance. So how accurate are your relative abundance estimates? So for this one, it's a little bit more nuanced because this could be measured in a bunch of different ways. So the first way is this L1 norm, and I give a formula right here, but it's probably easiest to describe with a graph. So basically what we have here is at a particular taxonomic rank, let's say the genus rank right here, we already know the true ground truth relative abundance of each of the individual genera represented with these blue dots right here. And then the predicted tool right here is going to make its predictions about what the relative abundances are. And then the difference between the prediction and the true, indicated here with the red lines right there, if you sum up the length of all these red lines, you get this L1 norm right here. So this is basically only going to tell you, give you results if your tool has actually predicted something as being present in the sample. Bray-Curtis metric is closely related to this. It's simply 1/2 the L1 norm to get the L1 norm on a 0-1 scale. But this L1 norm right here only gives us an idea of things that you've actually predicted to be in the sample. And what if you didn't get the actual genus correct, but you got a genus that was somewhat close to the one that was in the sample? How are we going to assess that? So we use this idea of a relative unifrac. And I give a definition right here, but it's probably easiest to describe with a picture. So in this picture that's probably way too small for you to see, I'll describe what's going on. We have the ground truth here decorating a taxonomic tree. And then we have the predictions of certain tool also decorating this taxonomic tree right here. The size of each one of these balls tells how much of the relative abundance was in the ground truth. And if it's split right down the middle, 50-50, like right here, it says that the tool predicted exactly the same amount as what is in the true right here. What the relative unifrac does is it says, well, how much of this blue stuff do I have to move around to get all the blue to overlap on all the orange right here in the most efficient fashion, keeping track of how much relative abundance you had to move and how far you had to move it over here. For the how far, the branch lengths, since this is a taxonomic tree and not a phylogenetic tree, the branch lengths is simply 1 over the depth in the tree. So using this, you can sum all these up, you get a number for the different metrics, and with relative unifrac values, you get some particular values. And you'll be able to see how this changes if we look at a better taxonomic prediction right over here. you can see that there's improvement in all of these metrics, showing an improvement in the abundance estimations here. As for the tools, these results are all computed with Opal, one of the manuscripts that Alice mentioned in the very beginning. We basically had 8 tools, a bunch of different methods. Some of them were Kamer-based, some of them use marker genes, some use alignment, some use machine learning, etc. And I'm mainly going to be looking at the marine data. So first off, looking at those binary metrics, just the presence absence right here, what I have are all the tools on these little spider plots right here, anonymized. So this is tool A, B, C, D, etc. Some of them were submitted with different parameter settings, but I simplified that for just this talk by picking one from each. And you can see that the completeness and purity, as expected, it decreases as you get to lower taxonomic ranks. But one interesting thing is if you compare it to the CAMI 1 results, which I have over here, yes, this is on different data, but in CAMI 1, we had this remarkable drop-off in these completeness and purity, or precision and recall, same thing, when going from the family to the genus level. All the tools just sort of fell on their face when they hit the genus and species level, save for just maybe one in precision. Thankfully, we don't actually see that for this particular class of data, for these marine data. We're doing just about as well at the genus level and instead of the family level as well, even though there's a slight drop off at the species level. Filtering is a very popular thing to do where you ignore very low abundance predictions. So at each rank, you can remove the lowest abundance taxa, summing to some arbitrary number. In this particular example, I'm going to remove the lowest 1%. And so here were all of my binary metrics that had no filtering applied. And then once we apply this 1% filtering, you'll notice that the results change. And interestingly, if you flip back and forth between these really quickly, you'll notice something really interesting. If you take a look at tool A sitting right up here, when you apply filtering, its purity jumps up a whole heck of a lot. On the contrary, if you look at tool B over here, you'll notice that when you apply the filtering, it actually hurts the purity of the tool. So while it might be common practice to do filtering, you got to really know how filtering is going to influence your particular tools, because some tools it helps, some tools it does not, as we can see right here. As for the abundance estimation, most tools struggle to get the exact abundance. So remember this Bray-Curtis distance related to the L1 norm. You had to get the actual exact taxa for it to be able to be counted. And I have noticed now that my screen has completely frozen. And I don't know if, oh, yep, still works. My mouse just does not work. There we go, we're back. And you can see that for here, the Bray-Curtis distance, larger is worse. And you can see that many tools struggle here to get the exact abundance of the exact predicted taxa. However, if you want to look at which ones are taxonomically close predictions, that's where the unifrac comes in. So looking at both unweighted and weighted unifrac over here, you'll notice some interesting results. So just to put these in context, lower is better, gold standard is 0. For the unweighted unifrac, good is something like a 0.01 kind of thing. So tools A, B, and C here are doing very poorly. But interestingly, for the weighted unifrac over here, we have, where lower is better, we have something really interesting going on here. This tool B, which is doing very poorly for unweighted, is doing quite well, actually the best out of all tools, for the weighted unifrac. And the reason for this is it has many low abundance false positives. Where on the contrary, if you compare this tool G, which does poorly in both unweighted and weighted here, that means it has many high abundance false positives. And you can go and make sure that this interpretation is accurate by actually digging into the data, looking at those taxonomic trees, zooming in on these, and you notice that yes, tool A is making extremely low abundance false positives, where this tool G is making higher abundance false positives. Strain madness, 2-second slide here. Basically, tools struggle on it when you have a lot of different strains, but interestingly, some tools did better on the strain madness data set than the marine, and conversely. So conclusions and takeaways here, there's a whole lot more to say. And actually there are some other talks that are more extended that you can go see these results in more detail. But basically the cool thing is that we have more tools performing more similarly, with a few clear winners and balanced winners between both binary metrics and the relative abundance. Filtering matters. Sometimes it helps, sometimes it hurts. And then abundance estimation is still challenging. Indeed, we have only one tool that is doing well at getting at least relatively close taxonomically to getting the predictions, and not many of the tools get the actual relative abundances done quite well. So there we go, 9 minutes, 41 seconds. Questions, I will be happy to answer any.
Thank you, David. That was wonderful. We've got a few questions on this one, some coming in the Zoom group chat as well as some coming in on the Q&A. So I'm going to take the ones from the Zoom group chat. In particular, Peter asks, what do you think the impact of the gain in reference sequences between CAMI1 and CAMI2 is on influencing the increase in accuracy? How much of that is the database?
Yeah, so that's actually really interesting. We are considering that question. We actually have some people working on that exact thing, which is using the old tools from CAMI1 with their old databases and running it on this new data right here to be able to measure that impact. And it just takes a long time to do that analysis. And so it's something that we are doing, but don't have any results on yet.